Okay. Good afternoon, everyone. Welcome to another edition of Café. Today, we have an eminent panel, and the topic is COVID-19, which is the hottest topic on the planet now. So COVID-19 and beyond. So it will be a discussion covering loads of varieties of topics from a mental health community point of view, etc. So I would like to hand over to and Dr. Arun, who will be facilitating this session. Can I please request that everybody goes on mute so we can hear the presenters? Okay, over to you, Suguchetan and Kishore Chetan.
നമസ്കാരം. പ്രിയ സുഹൃത്തുക്കളെ, ഇന്നത്തെ കലാകഫേ മീറ്റിംഗിൽ സൂം വഴിയും യൂട്യൂബ് വഴിയും പങ്കുചേരാനെത്തിയ നിങ്ങൾക്ക് എല്ലാവർക്കും എൻ്റെ ഹൃദയം നിറഞ്ഞ സ്വാഗതം. കേരള ആർട്സ് ആൻഡ് ലിറ്റററി അസോസിയേഷൻ കലാ UK ഈ സംരംഭം തുടങ്ങിയിട്ട് 10 മാസത്തോളമായി. പല സബ്ജക്റ്റുകളും ഞങ്ങൾ ഡിസ്കസ് ചെയ്യാറുണ്ട്. ഇന്നത്തെ സബ്ജക്റ്റ് COVID-19 ആൻഡ് ബിയോണ്ട് എന്നാണ് എടുത്തിരിക്കുന്നത്. ഇന്നത്തെ പ്രോഗ്രാം യുവതലമുറയ്ക്കും മലയാളം ശരിക്കും മനസ്സിലാകാത്തവർക്കും കൂടി കരുതി ആദ്യത്തെ പാർട്ട് ഇംഗ്ലീഷിലും പിന്നെ ഡിസ്കഷനിലേക്ക് കടക്കുമ്പോൾ ഇംഗ്ലീഷും മലയാളം കൂടെ ആക്കാമെന്നാണ് ഉദ്ദേശിച്ചിരിക്കുന്നത്. അതുകൊണ്ട് എല്ലാവർക്കും ട്യൂൺ ചെയ്യാം. അതൊന്ന്. പിന്നെ ഇന്നത്തെ പാനലിൽ എല്ലാ സ്ഥലത്തും കാണുന്നത് പോലെ ഒട്ടും കുറയാതെ ഏറ്റവും കഴിവും മികവുമറ്റ പേഴ്സണാലിറ്റീസാണ് ഡിസ്കഷൻ പാനലിലേക്ക് ജോയിൻ ചെയ്യുന്നത്. അതിന് വേണ്ടിയിട്ട് ഇന്നത്തെ മീറ്റിങ്ങിന് കോർഡിനേറ്റ് ചെയ്യുന്നത് ഡോക്ടർ അരുൺ കിഷോറാണ്. അരുൺ കിഷോർ കൺസൾട്ടൻറ്റ് സൈക്യാട്രിസ്റ്റ് ആൻഡ് എ ടീച്ചർ, കറൻ്റ്ലി വർക്കിംഗ് ഇൻ സസെക്സ് പാർട്ണർഷിപ്പ് ട്രസ്റ്റ്, NHS ട്രസ്റ്റ്. ഇത് നമ്മൾ തുടങ്ങുന്നതിന് മുമ്പ് ഒരു കാര്യം കൂടെ പറയാനുണ്ട്. ഈ കലയുടെ കോവിഡിനെ പറ്റിയുള്ള നിങ്ങളുടെ ചോദ്യങ്ങൾ എന്തെങ്കിലും ഉണ്ടെങ്കിൽ നിങ്ങൾ ശ്രീറാമിന് അയക്കുകയാണെങ്കിൽ മറുപടി പറയും. യൂട്യൂബിലുള്ളവർക്ക് ചോദ്യങ്ങളുണ്ടെങ്കിൽ നിങ്ങളുടെ കോൺടാക്റ്റ് വഴി ചോദ്യം അയച്ചു തരികയാണെങ്കിൽ വി വിൽ ആസ്ക് ദ പാനൽ ടു ആൻസർ യുവർ ക്വസ്റ്റ്യൻസ്. It doesn't matter what type of question it is, we are free to talk and we will enjoy the evening. ഒരു അനൗൺസ്മെന്റ് കൂടെ പറഞ്ഞോട്ടെ. ഇത് കഴിഞ്ഞിട്ട് നമ്മൾ തീർക്കുന്നതിന് മുമ്പ് ശ്രീ ഡോക്ടർ KJ യേശുദാസിൻ്റെ 81ാം പിറന്നാൾ ആഘോഷിച്ചുകൊണ്ടിരിക്കുന്ന സമയത്ത് നമ്മളും അദ്ദേഹത്തിനോട് ഒരു ആദരവ് കാണിക്കാൻ വേണ്ടിയിട്ട് ചെറിയൊരു കലയുടെ വകയായിട്ടൊരു സംരംഭം ഡോക്ടർ ബാബുരാജൻ കൂട്ടരും കൂടി ചെയ്യുന്നുണ്ട്. അതും കൂടെ എല്ലാ കാര്യത്തിലും ഈ മീറ്റിങ്ങിനും എല്ലാവരും സഹകരിച്ച് ഇതിനെ നല്ല ഒരു ഇതായിട്ട് കൊണ്ടുപോകണമെന്നുള്ള ഒരു അഭ്യർത്ഥനയോടുകൂടി നിങ്ങളോട് നന്ദിയും പറഞ്ഞുകൊണ്ട് ഇപ്പോഴേ നന്ദിയും പറയുന്നു, ഇനിയും കാണുന്ന ശേഷം പറയാം. അതോടുകൂടി നമുക്ക് തുടങ്ങാം. അപ്പം ആദ്യമായി ഞാൻ ഇത് തുടങ്ങാനായിട്ട് മോഡറേറ്ററായിട്ട് ആങ്കർമാനായിട്ട് ഡോക്ടർ അരുൺ കിഷോറിനെ ക്ഷണിക്കുന്നു. അരുൺ കിഷോർ. അരുൺ.
Hello everyone, and welcome to this panel discussion. Firstly, let me apologize for my slide, which is flipped mirror image. We'll try and change that in a while. But
no, it's okay. It's the same. It's flipped for you, but it's okay for everybody.
Okay, else. Wonderful. Thank you, Sridham. Before we start this panel, I'll just describe. People might ask or wonder why another COVID discussion. We've had so many, but this is unique in terms of expertise. And the focus today is on the UK and on India. And I think that's the important connection. It's a cross-cultural perspective. It is a professional perspective. It is a technical perspective. Now, when you watch, please remember that you might see the panelists discuss, sometimes differ in times about the nature of the issue. And that is the very nature of the problem, and hence we might disagree sometimes. Now, we'll start the discussion about the virus, talk about vaccines, and then go on to discuss its genetics and variants. We'll then touch upon epidemiology, especially the differences between countries, UK and India, and then a clinical perspective will follow. We have got people from different specialties: allergy, pulmonology, internal medicine, GP, and mental health. The format that we will follow is that each speaker will make a very brief presentation, and then we'll have a question to them from you and from other panelists, and then the panel will discuss. Now, please remember that you can send in your questions. You can either type them into the chat box or send them to Sridham or to myself. Finally, let me say that if anyone feels distressed because of the discussions, remember, you can always ask us for help. So let's start this panel discussion. Let's keep it live. Let's keep it exciting. Okay. Now, I'd like to introduce the first speaker, which is Professor Pradeep Vasudevan. Sridham, you just put up, yes. So Pradeep Vasudevan is a consultant clinical geneticist and honorary professor at the University Hospitals of Leicester. He has extensive experience, and he's a head of clinical genetic services at Leicester since 2010. He's also the visiting professor at Hubei University of Medicine, China. He's a chair of the clinical ethics committee, and that's something he will be talking about. He's a member of the special advisory committee of the Royal College of Physicians, and he's a training program director of the East Midlands. So he has various interests, as you can see in his slide. He's one of the senior editors of the newly commissioned book, Clinical Genetics at a Glance. His major research project is on deciphering developmental disorders, which is in collaboration with the Sanger Institute in Cambridge. And this has led to the discovery of more than 50 new genes, 280 complementary analysis, and 125 publications. We have an expert here. I'd like to welcome Dr. Pradeep Vasudevan. Pradeep, over to you.
Namaskaram. വളരെ സന്തോഷം ചേട്ട. Thank you for inviting me and for this such a wonderful panel discussion today. So I start by saying, actually I have to say only 4 main points. The first one is about the genetics of the virus. So I start by quoting Charles Darwin. I quote, so he is considered the father of evolution and natural selection. So I quote, "It is not the most intellectual of the species that survive, nor it is the most strongest of the species that survive, but the species that survives is the one which is able best to adapt and adjust to the changing environment in which it finds itself." And this is how I want to start, because this statement actually encompasses everything we are going to discuss today, both about the virus and about the humans also. So basically, it is an RNA virus. So we know that there are DNA and RNA viruses. This virus can infect a bacteria or plants or animals or humans. And we as a group in genetics and genomics have come together from all over the world and shared the data together. And this has led to the discovery of the vaccines around the world. So this collaboration is very important. And usually vaccine development takes about 4 to 5 years. That is the minimum time. But we, as a whole, as a human race, we've raced against time without cutting corners. We could actually finish the phase 3 trials of many vaccines. And there are many experts today who will be talking to you about various vaccines. So that's my first point about genetics and vaccines. The second point is how it has changed my clinical practice. So we used to have face-to-face clinics always, but since COVID has actually started from March 2020, everything is changed. About 70 to 80% of my consultations are now virtual. So we do almost all the consultations by telephone or by video. And this has changed the way we look at things also. We know that Kala Café is actually, as Mrna said, it is been 10 months. It's a product of circumstances and smart thinking. And at the same way, there are a lot of other ventures like Pallikudam and Bhagavad Gita classes, etc., which all came through because of COVID-19. And the power of IT here is so much. The third point I want to cover is about the mental health and emotional aspects. In my hospital, a lot of my junior doctors, I myself was not redeployed as such, but there were a lot of junior doctors and nurses who were redeployed and had to face a lot of difficult situations where they had to actually ring um, their relatives of patients who died and to break them the news, breaking bad news. And this has had a big emotional burden on health professionals and especially the frontline staff. And there is, for older people, there is isolation. There is many people have lost income, lost jobs. There is bereavements. And also for children, it's had a huge effect on the mental health and emotional issues of children. So I think that is a very important thing to consider also. And the last and most not least is the ethics behind it. So I'm the chair of the clinical ethics committee here at the hospital, and we had get a lot of ethical cases, so morally challenging cases where you have to maximize the resources and to limit the demand. How can we do that? And it has thrown up a lot of um, uh, difficult decisions we had to take. Who should be given ITU ventilation? Who should be given, mechanical ventilation, etc. So, so that is another point we would be discussing. So thank you very much for inviting me to this panel, and I think I'll stop here and we'll take questions. We'll be discussing more. Thank you, Cheta.
Thank you, Pradeep. That was brief and to the point and 4 very valid points. There was a question, I think, for you, which perhaps Sugueta, I think you were about to ask Pradeep a question.
The question was, could you briefly tell us about the mutations in COVID and its significance?
Okay, so thank you, Cheta. This is a very relevant question. It's a common question which is asked at different forums. Mutations are very common in viruses, and especially RNA viruses. I'm sure Veda Chetan will be saying more about this soon. So we already know that there are many thousands of mutations, which, but only a small minority are likely to be important to change the virus in an appreciable manner. So we have a big group in UK, which is known as COVID-19 Genomics Consortium. So we sequence all the samples from different ITUs, sample from patients, and we sequence it and put it out together. And we collaborate with other laboratories in the world. And like that, what we found was around October last year, 2020, we found a new variant. So there is a difference between a species and a variant. So this is just a change in a mutation, a changed a form of this particular virus. But what we have found, we have named it N501Y. So what that means is there is a change of amino acid, but it attaches itself to the ACE2 receptor very closely, and it increases the transmission of this virus. So this was found to spread very rapidly around London, especially Greater London, southeast and east of England. And this is the reason we saw such a huge spike of cases in December, sort of November, December, January. And this is exactly why we have the second wave, where we know that, every day we used to, the last 1 or 2 weeks, we've been having 50,000 people newly infected, and also more than 1,000 deaths per day. So it actually increases the transmission of this virus, but not the pathogenicity or mortality as such. because such a huge number of cases are coming at one point to the hospital, hospitals are also struggling, and that is a big problem now.
Deep, there is one more South African strain has been found, isn't it? Is it something similar to London one or something different?
It is actually different, Cheta. It's a different one. There are similarities, but it's slightly different. But now this is spread across the world. The UK variant is found in, it's almost about 100 countries. So whole of Europe and Far East, it's gone out.
I think we have a comment from Satyan talking about new variant detected in Southeast in September.
Sridham?
Pradeep, you're on mute.
Sorry, I think Satyan, you had a comment.
I just wanted to make a comment. This first variant, the one you're talking about, detected in September, not October. And unfortunately, it took time to get more details of that. So it's very obvious it was detected in September. And the news was broken only by probably early December. By the time it was too late. I'm personally talking about because I have personal experience of this new variant.
Mm.
Yeah, but Cheta, I think sometimes it happens is that even though the virus is, we get the sample in September, it might take about 8 weeks. There will be a gap before we can actually sequence and then publish it. So probably that might be the delay there.
Pradeep, there's a question from Sina, where Sina's asking you, why did the variant happen in the UK and not other parts of the world?
Well, there are, I think it is happening around the world. There are, but we are actually quite good in picking it up. And I think the Genomics Consortium UK actually sequences about half of the genomes of viruses around the world. So 50% of that it actually happens in UK, because we are really advanced in genomics, and that's exactly the reason why. I'm sure there will be different types of strains, like similar ones around the world, but they are probably not sequencing it. Yeah.
Thank you, Pradeep. I think we will move on to the next speaker, and we'll bring you back in the panel, Pradeep. So stay with us. So I now have pleasure in introducing the next speaker, Professor Mrna Gopal. Sridham, could you put up this slide? Yes. So Professor Mrna Gopal, he's given, I mean, he's OBE. Works in the Fulbright Institute in the Oxford University. He's a research scientist with major interest in viruses, and this is major interest. He's the head of the viral oncogenesis group at the Fulbright Institute, which, as most of you know, Oxford is where the new vaccine has been discovered. He will be talking about that. He's a visiting professor of virology, Department of Zoology in Oxford, and an investigator at the Jenner Institute in Oxford. Professor Mrna Gopal also has links with researchers in China, and he's actively involved in viral researches in the UK and China. So over to you, Mrna.
Thank you, Arun, for the introduction. It's a great pleasure for me to join today with the eminent panel from various parts of the UK and beyond. So I have a few slides so that I can introduce my research area. So I work at the Fulbright Institute, as Arun mentioned. So Fulbright Institute focuses mainly on animal viral diseases, and also what we call it as the human-animal interface, because it's becoming very, very clear that quite a lot of new viruses, such as COVID-19, emerges from animals. So you cannot simply study human medicine as one part of alone. We had to combine the animal also into the field. So this is called as the One Health area. So this is we focus quite a lot on animal viruses, but which has the potential to spread to the humans. Next slide, please, Sridham. but quite a lot of work which is relevant to COVID-19 is being carried out at the Jenner Institute in Oxford. So Jenner Institute, named after Edward Jenner, the father of vaccinology, who made the first vaccine way back in 1796. So this institute was started in 2000 with the whole idea of combining the expertise of various people in virology, immunology, and also vaccine manufacturing to come up with the sole idea of research into vaccinology. So we are a team of virologists, bacteriologists, immunologists. We work together, the sole idea of coming up with research into vaccines and also come up with the new vaccines. So we have been working for the last 20 years to develop vaccines for a variety of diseases, avian influenza, TB, malaria. There are a few examples. So Fulbright Institute, few of us from the Fulbright Institute is also part of this Jenner Institute, although our work is mainly focused on vaccines for the animal side. So when this COVID-19 came about in early January, my colleague Sarah Gilbert, working in the Jenner Institute, took up the initiative of developing a vaccine. And then again with my colleague Andy Pollard, who is a pediatrician working on vaccine development. So that's how the whole thing came about. Next slide, please. And as Pradeep mentioned, it was a record time that we could develop the vaccine. So I was in India in December in 2019 when the news about the COVID-19 came about from Wuhan. An unusual respiratory syndrome was happening there. And Wuhan Virology Institute, with which I collaborate, they came up with the first sequence and made it available to the whole world in early January. And as soon as we got the sequence, the Jenner Institute started working on developing the vaccine. The vaccine was made in record time. In less than 3.5 months, it went to the trial. This was because we have been developing a number of vaccines, and quite a lot of the vaccines are made using a platform called as the adenovirus vector technology. So we know quite a lot about the adenovirus vector platform. So it doesn't matter which vaccine or which vaccine against which disease you want to make use of. You can use this platform very quickly, and we can grow this virus and produce the vaccines in a very, very fast time. And also we have the facility for initial testing. So that is one of the reasons why we were able to have the vaccines developed very quickly. Next slide, please. And so today, as many of you know, we have 3 vaccines approved by the MHRA for use in the UK. The first one is the Oxford AstraZeneca vaccine. So, which is most of the vaccine is actually made in Oxford, in Cowley, in the Oxford Biomedica. Also, we have another partner at Cobra Biologics in Kiel. So these are the 2 major sources where the vaccines are actually made, and it's bottled in Wrexham in the Wockhardt facility. So today is a very important landmark in the vaccine in the COVID-19 in the UK, because today, if you look at the records, we have surpassed the number of cases. So we had totally about 3.3 million cases of COVID, and today we have done 3.5 individuals have been vaccinated. So we have overtaken the number of cases we have, and also we are vaccinating at a rate about 4 times more than the new cases that are emerging. So I think we are winning the battle here. So, this is where my work comes in. But since we are talking today about the COVID and beyond, I thought I will touch on what can happen after this, whether we can all relax and sit quietly. The next slide, please. So there are quite a lot of diseases. So if you look at the last 60, 70 years, we had about 400 new infectious diseases coming up. Some of them cause severe disease, some of them go unnoticed. So nearly 75% of them are zoonotic, are coming from animals. So we will continue to have these sort of diseases, and COVID-19 may not be the last one. So one of the things I think we should consider beyond the present pandemic is that how are we going to deal with the future pandemics? So the next slide, please. So for that, we had to understand where are these diseases coming from. Quite a lot of the diseases are coming from the bats. So bats are the kind of reservoir of this, quite a lot of the viruses, the SARS in 2003, the MERS in 2014, and the COVID-19, these are the coronaviruses. All of them actually came from bats, although other species of animals are also involved. And in addition, we have the Marburg, the Ebola, in the Nipah virus, which unfortunately killed 17 people in Kerala, also came from bats. So how are we going to deal with these potential danger lurking in the bat population as a reservoir? So this is something we have to seriously consider. And the next slide, please. And also, if you look at the world, so this is actually a real-time image showing the number of aircraft in the skies, 24/7. So any virus which comes from a bat or any animal species jumping into humans in any corner of the world can potentially result in a pandemic. So this is something of a huge concern. So in Oxford, we have been seriously discussing about this, what we're going to do about this. And yeah, so one of the ideas which has been put forward is to have a center for pandemic preparedness. So we have a very active program. So we will be starting a center which is a multidisciplinary approach with a lot of collaborators around the world. So rather than waiting for the virus in the bats or the animals to catch us, we want to go and catch them first. So it is estimated that there are something like 5,000 potential viruses are in this population of bats. So with the advanced genome sequencing technology, the satellite imaging, quite a lot of new technologies are potentially available for us to look at hunting these viruses. So this is where I think quite a lot of research and effort will be going on in the future. In 6 months, 1 year time, once the dust settle down from the COVID-19 pandemic, we will be actively seeking to have active research so that we can catch these viruses and be prepared. So once we have the viral sequences, we can start developing vaccines or antiviral drugs and keep it in our armory so that when these guys come about, we can deal with it quickly. So this is something which we all should be thinking about, because this COVID-19 definitely, in my view, also will not be the last. Thank you.
Thank you so much, Mrna. I think you've sort of given perhaps me and perhaps the rest of the audience a very positive note there, because I took two lessons from what you were saying. The world has opened up, but the world has opened up in that scientific cooperation is what developed the vaccine for the virus. So, knowledge is no longer compartmentalized. The second, I think, important point you raised was about the future. I mean, we need future preparedness. And you've started it by saying that we need to develop vaccines, we need to develop medicines for future infection. And I wonder how the rest of the world and the other specialties are going to be prepared for another pandemic as well. But I think there was a question for you. You were talking about animals. I think there's a question for you about animals. Sridham, do you want to ask that question? Sridham?
Yeah, so I was in the background. I was checking a few things. So yeah, you talked about testing these vaccines in animals. What is the effect of the of this COVID virus on animals and pets? We live with these animals. So how worried should we be about our animals?
Yeah, I think that's one of the earliest thoughts we had as soon as we had the outbreak. We wanted to look at the wide spectrum of hosts which can be affected by these animals. So I think Pradeep touched on this. So the virus entered the cell through a specific receptor called the ACE2 receptor. And so fortunately, at least in the initial stages of the study, quite a lot of the ACE2 receptors are not used, the right sort of ACE2 receptors are not present in many of the domestic animals like cattle, sheep, pigs, chickens. So they don't have the receptor. So at this stage, we do not think that the COVID-19 per se is a big risk to quite a lot of the domestic animals. But having said that, there have been cases of the disease in dogs and cats because they still can be infected, although we haven't seen major outbreaks. So there have been mortalities in lion. A couple of days ago, there was a news about the disease in chimpanzees. So the virus can gain into that. And we saw in the Netherlands, there's a huge escape of the virus into the mink population, the farm mink population, and a large number of them have to be killed because of that. So this is particularly happening when the virus mutates. I think we mentioned about the mutations in the viruses. So technically, the original virus have a very narrow restricted tropism for the humans, but as the viruses potentially mutates, they gain the capacity for the changing the tropism. So there's always the potential for that, but at the moment, I think the risk for this disease to animals are restricted to, I think, the pets like the dogs, cats, perhaps the primates like the chimpanzees, etc. But there is always the danger, and so it's all the more important again we have to contain the virus. So as and when the virus replicates more and more, you'll have more mutants coming in, and then they can establish into, or they can expand their tropism and expand into other populations. So at the moment, I think they have a reasonably narrow tropism, but it is probably not the end of the world. I mean, maybe you can possibly get new tropisms and it can spread into animals.
Okay, thank you.
Thank you, Venu. Now, there's a specific question to you and to Pradeep, and this is from Hema Santosh. It says, what are the chances of the adenovirus reactivation in the Oxford vaccine in the immunocompromised?
Yeah, that's a good question and a concern for lots of people. So first of all, the adenovirus which we are using is a reasonably harmless virus. It's a virus which commonly causes cold. And on the top of it, these are all has been kind of disabled viruses. So it means the adenovirus require a couple of genes, absolutely, called E1, E2, E3, E6. There are so many genes. So we have deleted from this virus the two genes, important ones, E1 and the E4. So once you delete these genes, they are disabled. They cannot replicate at all. So what we are giving as vaccines in the humans are disabled adenoviruses, which will deliver the antigen into the cells, and thereafter they cannot replicate. They are just a vector to deliver it. They do not replicate. If they were replicating in the vaccine recipients, definitely there are questions like reactivation, immunocompromised people, etc. But at the moment, these are disabled adenoviruses. They just are delivered there just as a vector, as a vehicle for delivering the antigen. So the question of reactivation, etc., doesn't arise. But on the top of that, this vector, which has been used for the last 15 years in several vaccine trials, like even influenza vaccines in elderly people. Quite a lot of people have been given this sort of adenovirus vector influenza vaccine, Rift Valley, tuberculosis, malaria, even pediatric malaria, 3-month-old babies have been given. This is an extremely safe system of delivery. So we don't expect, and we haven't seen anything untoward so far, except for some mild reactions like temperature rise, etc.
Yeah, I have the same thing to say, Cheta. That is, I fully agree with what Venuchutan said. This is a tried and tested method, the Oxford vaccine. It has been used for more than 25 years to deliver the vaccine to the host cell. This is known as ChAd-Y25. This is a particular adenovirus gene, adenovirus which is used, which causes common cold in chimpanzee, but it doesn't. It's actually disabled for humans. So I don't think it will cause a problem, because we have used the same technique for the flu jabs, which we do it every year. So this is exactly the same technique. So it's a tried and tested method for Oxford vaccine. Yes.
Thank you both. That's very reassuring. One quick question, which is about, do you have a preference for the Pfizer or the Oxford vaccine in immunosuppressed patients?
Well, okay, Chetan.
Yeah, I'm not an expert to sort of compare, but all the regulatory authorities, MHRA or FDA or whoever looked at all the vaccines have given approval. So I don't think there is any preference. All of the vaccines are doing the same thing. They're delivering the spike protein, which is the most immunogenic protein, in different forms. So adenovirus vector is using the adenovirus. It will be delivered and it will produce a protein, whereas the other two vaccines, the Pfizer and the other vaccines, are RNA, which will be also delivered, which will be made into a spike protein in the system. So I think it's essentially the same. So I don't see much difference in terms of the immune response, although the initial studies are showing some differences in the protection, but that may change as we have more and more samples, vaccinations being done.
The other important point to think about is also the genetics of the immunocompromised patient who is being given, the medicine. It's not just the virus, the vaccines genomes, but it's also the genomes of the whoever it is given to. Theoretically speaking, mRNA vaccines, which is the Pfizer and Moderna vaccine. I'm sure Shyam will be able to say more about this, but it's the half-life is very low. So once it comes in, if you theoretically speaking, it just disintegrates. So the mRNA makes the protein, and the mRNA will disintegrate in the cell. It doesn't use the cell sort of genome machinery like that. So I do not think it's a major issue. Shyam, you want to come in?
Shyam?
Yeah, so the mRNA vaccine usually, mRNA stands for messenger RNA. So these are the small piece of RNA which has the codes for a part of spike protein. It's not the entire spike protein. To make a good immune response, you need only a really small part of spike protein, which is the most immunogenic part of the spike protein. So the way they have formulated that is this small mRNA is covered in a fat layer called lipid. And once that's injected intramuscularly to our body, this mRNA will translate, or using our cell machinery, it doesn't use any of the genetic material, but just use our cell machinery, it translates the mRNA to small bits of proteins. So then you see, then our body will develop like small spike protein, bits and pieces of the spike protein. And that spike protein should, or it will trigger the immune system of our body and makes a lot of T cells and B cells. So the T cells basically kill the virus, and the B cells produce a lot of antibodies, neutralizing antibodies, as well as memory cells, etc. So that's the that's how the mRNA works. And as Pradeep said, this mRNA, it doesn't cling in our body. It only stays for maybe a few hours, and then it get destroyed by our by itself. So the mRNA doesn't integrate or anything. So that's the beauty of this new technology called the mRNA vaccine, and that's pretty fast too.
Thank you, Shyam. I'll apologize for bringing you in without any introduction at all. This is the right time to introduce you.
No problem at all.
This is the right time to introduce you. So here you've already seen Dr. Shyam Nambole, and we're very grateful to have you here. He's actually in Pittsburgh, USA at the moment. So Shyam, welcome to our panel discussion. Shyam is working as a SARS-CoV since February 2020, and he worked on the pathogenesis, animal modeling, development of a therapeutic agent in the vaccine study in different animal models. He is from Parenur. Shyam studied masters from the London School of Hygiene and Tropical Medicine, the University of London. He completed his PhD in virology from the Queen's University, Belfast, and worked as a clinical virologist in the NHS in Glasgow. So he's homegrown in all ways. He moved to the USA, we lost him, in 2012, and since then has been working as a research scientist in the virology research lab there. So over to you, Shyam.
Thank you very much. First of all, I want to thank all the panel members to invite me to this eminent panel. So as Dr. Arun said, I studied in UK, my master's and my PhD, and then I moved to US. And in the US, I mostly got trained as like BSL-4 pathogen. I was not working in a vaccine lab. I was more working in a research lab. And end of 2018, we moved to Pittsburgh when my boss, Professor Paul Dupre, he's the director of the Center for Vaccine Research. And since then, I've been working on vaccines. So whenever I said vaccine, doesn't mean that we actually make a vaccine or like, it's not a vaccine production. It's more of vaccine research. Like you go right to the virus, make a disease model, and then find out how to actually make a vaccine for it. So that's exactly what I did with coronavirus when we started working with coronavirus from February of 2009 and 2020. So first we started finding out a best animal model to do to see the infection happening. Like we tried ferrets, monkeys. In monkey itself, we have used 2 different species, macaques and African green monkeys, and we found that African green monkeys are better, and we already published all that data. And then we have decided to do a vaccine development, and we used measles as the vector, and we used the measles vaccine and then incorporated the entire S gene. So basically we cut out the gene, the S gene from the coronavirus, and inserted that into a measles virus, or measles vaccine virus, not the measles wild type virus. So that's called recombination technology. And once we develop that, we have a collaboration with Serum Institute of India in Pune. So they are the one who formulated this vaccine for us. So they put it in a bottle and then make it as like a, add all adjuvants and whatsoever, and then send us back. And we tested that in 2 different animal models. We tested that in mice and also in monkeys. And both of them showed very good results. As of now, we got like good immune response. There was neutralizing antibodies, and there was disease, the lung pathology was much different from the vaccinated group from when you compared with the control group. And on top of that, I also work on developing some therapeutics with some structure biology people in University of Pittsburgh. So this one, we work as something called nanobodies. They are basically antibodies or antibodies. So these nanobodies are antibodies which are seen in camel and related species like camel, llamas, and all desert animals like that. So we actually injected the spike protein in llamas, and we took their blood. We extracted blood, we draw blood, and then we took the serum out of the blood, and there was nanobodies, which is like antibodies of our body. And then we extracted that. We did like a big study, which is the best nanobodies which can go and bind the SARS coronavirus. And right now I'm working on that, and I'm using the nanobodies in hamster models, which are another animal model, which is one of the best model, I would say, better than monkeys, to be honest, whenever you want to see the disease progression, because in hamsters, the disease progression of coronavirus is pretty good, especially the lung involvement of the SARS coronavirus. So in that, we actually got a pretty good result. So I'm trying to publish that in a journal soon, but we already published the nanobody
settings.
Sorry, we already published the nanobody technology in Science Journal sometime in the fall. So hopefully this one also get published. And whenever we're talking about the variants, so we did have a interesting case found out, and that's just in a journal soon. We just got the revision, and we resubmitted it. So this is like a this was a case study. So there's 2 sections of the study. One was published in a Journal of Clinical Disease. So this is a cancer patient who was infected with SARS coronavirus sometime in spring 2020, and then he had a really prolonged disease. He was like a coronavirus positive for 72 days. So it's like a very persistent infection. And on day 72, the doctors collected the ETA, the endotracheal aspirate, which is an aspirate from the lower respiratory tract. And I isolated the virus, and then I sequenced it. And then whenever I sequenced the S gene, we found out that there are 2 amino acid deletion. And then these deletions are very, very important. This is one of the neutralizing antibody escape. So this is like an adaptation of the virus, how virus is so clever, how it can invade our immune system. That's what we have seen. And then we are trying to publish this again in Science Journal. So I found this like super duper interesting, and we are working more on that, the mechanism and other things. So that's an ongoing study.
Thank you so much, Shyam. You sound so passionate about what you do. I think you possibly raise the passions in a lot of people and their interest. So perhaps there's a question that you could answer, Shyam. This is from Satyan, and Satyan is asking a question. Do you anticipate any changes or requirements for tweaking the vaccine in future to cover multiple mutations?
That's a very interesting question, and it's a little difficult question too. I would say that most probably yes. The way it's going, let that be the UK variant, and whatever we have seen. Like I've just told you that I have seen a virus which can invade some of the neutralizing antibodies, but there is still. Remember that this is a really new virus, and there is so much need to be learned. We all think that in the initial days, we all thought that the there is something called RBD, which is the receptor binding site, whereas the spike protein, the reason why the spike protein is so important is spike protein is the protein where the virus facilitate or our cell will facilitate the entry. So we have the ACE2 here, and this is the spike protein. It's basically literally come and sit like that. And then that's how the virus enter to our cell. If this is not happening, the virus cannot enter to our cell. So this is why the spike protein is everything. Whatever the vaccine we are making, everything is depending on the spike protein, because all the antibodies will come and bind, and it will stop this binding. So the reason, the way the spike protein or the coronavirus evades this immune response, our immune response is that by tweaking, sometimes the protein is like this, and then it will, by the mutation or deletion, it changes. What do I say? The structure of the protein slightly changes, and therefore the antibody cannot bind. So we are started seeing that. This is one of the report which we have published. It's one of the first report, but I've seen that there are more reports coming out. So the more we study, and the more the virus is like circulating, and this is the time, because this is the peak of the infection happening now, and this is the time virus adapts. The virus adapt to our immune system, and all these changes is going to happen, or we will see it only in another few months. And the more we will see it, then we have to answer your question, like, do we need another vaccine? We might need it. Who knows?
Just adding one more thing, Cheta, that is, it's a difficult question to answer, but clearly there will be more mutations or in the future. And for influenza vaccines, every year which we take in the NHS, the jabs are tweaked depending on which is the dominant strain that year. Yeah. So similarly, there is a good possibility that this will happen for the SARS-CoV-2 also. There will be mutations in the future, and the vaccines may have to be tweaked for it to be effective. But for this consortium and the genomic sequencing of various viruses has to happen quite regularly, and the data has to be shared to see which is the dominant one.
Yeah. So to adding to that is in the flu, it's more of like antigenic drift. It's basically changed the bits of the genetic material, like moving it, and things like that. Whereas in coronavirus, it's more of like mutation. So this is very unpredictable as of now, what's going to happen, or there are deletions as well. So the deletions is also very, very critical. So in that way, we just need to wait and see what is coming up, and depending on that, we need to change or tweak the vaccine. That's what I think.
There are lots of questions on the YouTube, but I think those questions I will ask once we progress a little bit more. This is a question perhaps you can answer, Shyam. And this is from Radhakrishnan, and he asks, the mRNA technique seems to be very interesting. How standardized is the computational biology so that new vaccines can be created by pressing a button and running the program?
So, of course, the technology has definitely helped. That's the reason why we have an mRNA vaccine, which is less than a year time now. Let that be, the bioinformatics and the computation biology has really helped analyzing all these huge number of the sequences is nothing but the genetic information of the virus. So we know about this deletion, we know about this mutation, and all that's only by the technology we have developed all these days. But then being said that, just because we have a technology also doesn't mean that we can have like a vaccine. It's a combination of biology with computation. So it's like a teamwork. And also, someone has said previously, the one thing which we have learned, is that, as I said, never in history we had like a vaccine in less than a year. Usually, the vaccine, by the time, at least in USA, by the time we get like FDA approval, it's usually take like 10 years at least. And I think one of the fastest vaccine previously was mumps, if I'm not wrong. So, the way the technology has developed and how the pandemic has made us to actually push it to make a vaccine less than a year is just like amazing.
It is. Thank you for that. There's another quick question to you, Shyam, which is, and this is from Santosh, can tweaking the vaccine also mean that a cocktail for multiple variants? Can you have a cocktail of vaccines from all the variants?
That's a really, really good question. And that could be one of the way we probably will do it in future. But this is, again, it's such a difficult thing to say right now, because these are things which are really studying. Maybe Pradeep can actually join in as well. I would say, because this is something, as far as I know, I think it's so difficult to actually predict that this is going to happen. It is one of the way we should think about, this probably will work. And the thing with vaccine is, many of the thing we'll think that, in vitro in the sense like in the lab, we'll think that, this is going to work amazing. And then whenever we go to a phase 2, the efficacy of the vaccine is pretty low, and it never going to see the market. And this happens all the time. So these are things which is like open up, and this is why we should do. Many different people are doing so many different way of making a vaccine, and I don't think that nothing is waste. Like, we are making a live attenuated vaccine, and since there is a Moderna vaccine and a Pfizer vaccine, and there is another AstraZeneca or Oxford vaccine, it doesn't mean that we have to stop and go and continue with something else, because as we all know that we always need a better vaccine, even though there is a vaccine available, and you never know, because these all of RNA viruses, as Pradeep and Dr. Venu said previously, RNA vaccines keep on mutate. And especially some vaccines are worse than some viruses are worse than the other.
Sorry, adding, and also, thank you, Shyam. Adding to Santosh's question and answer is, this new variant, which we have seen in the southeast of England, has mutations to the spike protein that the 3 leading vaccines are targeting. But however, the vaccines produce antibodies against many regions in the spike protein. So it's unlikely that a single change would make the vaccine less effective. So I would say we can tweak the vaccine, definitely.
Yeah. So
I don't know whether that is clear, Santosh.
Yeah, just for your information, Pradeep, the case which I was talking about, the deletion, we have seen that deletion in the NTD, which is the N-terminal domain. Just to let you know.
Thank you all for that detailed answer. This might be the right time for us to then bring in the next speaker. So do stay with us, Shyam, and we'll bring you back for the panel.
Sure.
So I have great pleasure in introducing the next speaker, Dr. Pratap Pillai. Pratap is going to come up. Hi, Pratap. So Dr. Pratap Pillai is a respiratory immunology consultant and an allergist. He's at Guy's and St. Thomas's NHS Foundation in London, United Kingdom. And there'll be a lot of questions to you, and I can already see questions to you come up on allergies with the vaccine. So, but I'll let you go first. It's all yours, Pratap.
Thank you, Arun. Thank you for your kind introduction, and thanks to Kala and of course Subichetan for inviting me to be part of this panel. And yeah, we heard about extensively and in detail about the genetics and the various aspects of COVID virus and touching upon various aspects of vaccine. So I would like to do a very brief introduction. I'm kind of slightly sidestepping from what you've been talking and perhaps touch upon something very slightly different, slightly more clinical, and perhaps interesting to many people. I hope so. Now, the first part would be what Pradeep said, how COVID has affected my area of work like anyone else. And the second part, I would try to touch upon something about the allergic reactions to vaccine from my point of view, in the context of allergies. So we all know that actually, COVID-19 has affected our life and how we interact and live and socially or from the work point of view in the past, say 12 months or more. Yeah, it has affected all of us. In a similar way, it has affected my area of work also. Allergy department, I work as an allergist, a clinical allergist at Guy's and St. Thomas's. It's a very narrow and specialized area where we see people with different kinds of allergies. And my work area was mainly based on outpatient clinics and day unit activities rather than inpatients or on-call works and all that. So like in any other walks of life and services or organization or business, my specialty was also affected. So, and there were a lot of restrictions and restraints imposed due to the COVID in terms of manpower. For example, sickness and many people calling off sick and going away, not available for work. I'm sure that you all have experienced the same thing. And in terms of hospital space, actually, because when a big pandemic comes in that brings in patients with a lot of other major emergencies, probably the priorities change. They need space. You have seen in the first wave of COVID, actually, so how they occupy. The hospital beds and the ICU beds were not sufficient. So they're looking for any spaces in the hospital to keep patients in, to admit patients in. So in that way, the space was affected. So that was another restriction. Of course, like in any other businesses, funding was affected. And many services were cut down. Actually, you won't be having funding for the next 12 months. We will reconsider in the next financial year. So in many ways, service was affected. And that is a common platform for all of us, actually. I think I'm sure that all of you have faced that. Like Pradeep mentioned, actually, we were also, it's interesting, actually. So prior to COVID, until January or February 2019, I used to do all 100% face-to-face clinical activities. And many a times the management used to come and ask us, can you do a little bit of telephone clinics and virtual clinics or Skype clinics? We all used to say, oh, no, we need to see patients. We need to touch patients and feel patients to understand, and etc. So always, this conversation went on. And what happened now? So that's interesting to see that, actually. Roughly around 60% or 70% of our clinical activities are virtual, either over telephone or Skype and whatnot. So I would like to show something in that context. Actually, I'm going to open a slide here. Right. Can I check? Can you see my computer screen?
Not as yet.
Not as yet. So I'll have to go on and share screen. Can you now?
Yes.
Yes. Yeah. Give me a second. Sorry about this. Yeah. Right. Okay. So, right. So I mean, some of my colleagues had looked at objectively into it and see that what was the difference and came out with this kind of. I mean, we were able to look at. See, you can see that to describe the outcomes of the use of synchronous telemedicine, essentially telephone appointment for the outpatient consultation in our center. In a month, we saw 637 non-face-to-face appointments in April 2020. And we came out with the conclusion that this is a very attractive service model, and a very time and cost effective, can improve the patient access to specialist care. Just to bring in that context, actually. So how we provide services has changed dramatically over the period of last 1 year. Now, I'm kind of moving on to the second part, what I want to introduce to you. And we heard about the vaccines, various vaccines which are available. I'm sure that someone else will be talking on that. And from my point of view, or from my point of view, as far as allergy reactions to vaccine are concerned, I just want to introduce this concept into you. Who should not receive the vaccine and get advice? And fortunately, I have to tell you that there are only very few individuals who cannot receive the two vaccines which are currently in circulation, which are in the UK from an allergy point of view. And anyone who has had a very serious allergic reaction to a previous dose of one of the COVID-19 vaccine or any component or excipient of the COVID-19 vaccine. Now, I'm sure that you might have heard about the initial, after having received the first batch of Pfizer-BioNTech vaccine, a handful of patients, actually, very few patients had developed severe allergic reactions. And after having brains into it, after thinking about it, the assumption is that one of the ingredients of excipient called polyethylene glycol, which is from a group of common allergens found in this polyethylene glycol, is a substance which can be present in vaccines, in medicines, and various other household goods and cosmetics. So if anyone is aware that they are allergic to polyethylene glycol, they should get an advice before receiving the vaccine. As you can see, what I have put there, this allergic reaction to polyethylene glycol is extremely rare. But if you know that you're allergic to polyethylene glycol, if you're sure that if you react to that, you have to get advice. And if you don't know, there is another condition that actually which may raise an alarm, which is that if you have unexplained severe allergic reaction to many different medications or other things, probably that is that also should raise an alarm, and you should get advice. So that is one of the part of the current guidance from the MHRA as well as British Society for Allergy and Clinical Immunology, that if you are known to be allergic to multiple classes of drugs and had severe allergic reaction, you should not be vaccinated with the Pfizer-BioNTech, which is known to contain this ingredient as an excipient. And if not otherwise, if you don't have other problems with the AstraZeneca vaccine, it can be considered as an alternative. And if a person had a reaction after vaccination, like a lumpy, itchy skin rash to the first dose of COVID-19 vaccine, he can still receive the second dose of vaccine with a little more supervision, a clinical supervision, perhaps in a hospital, and a longer observation time. So these are the important points I would like to highlight. in my brief introduction, and I'm quite happy to answer any further questions regarding this. I'm just going to stop. Okay. And thank you very much. Stop share. Have I stopped sharing?
Yes, you have. Thank you, Pratap. And I think you've sort of brought the virus into human contact now. We were talking about virus in isolation. We were talking about various types of vaccines, but you brought the human element in here, and I think that's important. So one of the questions, and this is something from India, which is the death of an elderly person in India after having received the vaccine. Should the elderly be careful or more careful here?
Well, I mean, from my context, actually, from allergy point of view, any person who is receiving the vaccine is, I mean, that's a different, I mean, I haven't read anywhere that the elderly should be particularly careful about the vaccine unless he has got some kind of background problems, as I have mentioned. But there are other aspects in which you have to be careful in giving the vaccine. But that is, I don't think that is part of allergy as a thing. Actually, we have to be careful. And a detailed guidance on and it's available from the vaccine manufacturers and various regulatory bodies to go through. For example, UK, if you have any doubt, actually, you can read the book called Green Book, published by Vaccination Group. Actually, there is a regulatory organization here, which gives a detailed and a beautiful description about where you have to be careful and discuss further about before injecting a patient with vaccine. And I cannot say that, actually, that's incorrect to say that about 60 years of age, about 70 years of, you got to be careful. That's a bit of generalized term. So because those patients need their vaccination, actually. I mean, if at all possible, you should give the vaccine unless it is absolutely not possible to give the vaccine. That is my view on take on this.
Shyam, did you have anything to add to that?
No, I was actually surprised because in both in US and UK, as far as I know, senior citizens like above 70 and 80 are considered as the prime priority for this vaccination, so that doesn't, all these one-off cases always there with any vaccination or any even antibiotics, some of the patients get really adverse reactions, so that we cannot generalize that.
There's a question about, somebody with a penicillin allergy who's refused the vaccine. What is the alternative for them then?
Arun, that's an interesting question. I saw the questions on my side panel that came from Sina, and there was another person also the question. And that is, I was anticipating that question because possibly it was our vaccination program started, I think, if I'm right, it's in the first week of December, right? So in the first 2 weeks, actually, there were a few major reactions reported with this Pfizer vaccine. And suddenly, they didn't know what exactly was happening, 5 or 6 patients with vaccine. And naturally, if there is any serious adverse reaction to any medicinal product, it would be reported to an organization in the UK called MHRA. So MHRA reviewed these reports and said that any person with a previous history of serious allergic reaction to a food, medicine, or vaccine should not be given the vaccine. That was the first decision MHRA took. So it was it raised a lot of confusion in the first few days of vaccination. And then our kind of organization, British Society for Allergy and Clinical Immunology, had to have a series of meetings and discuss with the MHRA. And fortunately, towards the end of December or early New Year, they amended that. They amended that decision to say that only if you had, that's what I have shown in my slide, actually. According to the current guidelines, any person, you should not receive a vaccine if you had a serious reaction or anaphylaxis to a serious allergic reaction to the first dose of vaccine, or if you're a person with multiple serious allergies to a different group of medication or an unknown. We don't know what you're allergic to, but you get repeated episodes of anaphylaxis. ? You're allergic to PEG. In that case, you should be referred to like our department to see that what can be offered to that person. It needs to be investigated whether this person is allergic to any particular ingredient or excipient of the vaccine. That's a fact. So that happened. There were many patients in our hospital also were refused vaccination. We used to get calls day in and day out about this, actually, whether this patient is allergic to penicillin, had allergic reaction to penicillin 30 years ago. And that poor lady is sitting there actually with various other problems, and she will benefit, definitely would benefit from vaccination. Whether can we give vaccine from the person at the vaccination port, and we have to go through all the details and say that, okay, go ahead, vaccinate that lady. So that has been reviewed. Now it is easier. Now this person can receive the vaccination, because if a person is allergic to penicillin, that doesn't preclude that person receiving the vaccine.
Thank you, Pratap. And thank you, Sina, for asking that question. Sorry, I didn't mention your name. There's another question from Malini Vishwam, who asked the same sort of a question, that her son carries an EpiPen and they don't know what he might be allergic to. And what would your advice be?
Exactly. There are both the cases, actually, both the contexts which I have discussed just now in these two questions. One is penicillin allergy, one is carrying an EpiPen for an unknown agent. That may need a bit of discussion. For example, there's a remote 1 in a million chance that this person is allergic to polyethylene glycol, who knows? In that case, the person can choose a different vaccine appropriately if it gets referred to a proper, a right person in the hospital specialist.
Thank you, Pratap, for your patience. Please do stay back for the rest of the panel. I'll bring in the next speaker now.
Thank you.
So I'd like to call in Suresh Menon, Dr. Suresh Menon.
I think he needs to start his video before I can add him.
Suresh, could you start your video, please?
Yeah.
Thank you, Suresh. So I have pleasure in introducing Dr. Suresh Menon. So he is a consultant anesthetist in Milton Keynes University Hospital. So he's an NHS clinician. He's a founder member of the Buckingham Medical School, new medical schools coming up in the UK. So Suresh is a founder member of that. So he's a medical educator. He studied in Thrissur Medical College. So he's homegrown, and he's the president of the Milton Keynes Malayali Association. Welcome, Suresh. Over to you.
Thank you, sir. It is indeed a great pleasure to be, what I say, to be hosted by my teacher. Arun Kishore sir was our psychiatry teacher in Thrissur Medical College, and Gafur is sitting here. So me and Gafur are classmates. And Arun Kishore sir, we remember in late 80s, was teaching psychiatry in Thrissur Medical College. And I think the next time I met him was actually here in UK in one of the Kala events a few years back. So thank you very much. Thank you, Kala, for inviting me to be in the panel. And I haven't got any slides. So what I will actually do is, I work as a consultant anesthetist and intensivist in Milton Keynes University Hospital. So what I will sort of do is maybe quickly look, do our experience in Milton Keynes. I have got actually a few numbers. I'll just quickly tell a few numbers, what has happened in the first peak, what has happened in the second peak, and maybe take some questions rather than actually going through any slides. So what we have seen is actually the second peak compared to the first peak. Definitely, I think we have more patients. And as any hospital in UK, majority of the intensive care units are full. So Milton Keynes, it is not actually that bad compared to London. If you compare to London, a lot of the intensive care units are running at like 150%, 200%. Some of them are even at 250% capacity. That means if an average intensive care unit in UK has got around 7 to 10 intensive care beds. So I will just give you an example. Milton Keynes has got a population of around 250,000, and we have got around 7 intensive care beds. So that's what we have. So roughly around 3 beds per 100,000 population. That is roughly an NHS sort of number. And in winters, in any UK hospital, in winter, the bed occupancy is very high. It can go 90%, it can go up to 100%. Sometimes we have to transfer patients because we don't have a bed, we transfer to another unit. But in the first peak of the COVID, all the hospitals had plans for COVID surge. So we had capacities built outside the main unit. Usually a recovery, the post-surgical recovery unit was converted as an intensive care unit. And that was used as an extra capacity for the intensive care beds. Luckily in Milton Keynes, we didn't have any sort of necessity for that sort of facility being used. So we were roughly running at around 10 to 12 patients throughout the first peak. And in the second peak, now what we see is basically in the hospital, we have around 200 patients, and we have got around 10 or 12 patients in the intensive care unit. So the capacity is not, we are not sort of, like any other London hospital. So we are actually doing okay. But if I actually look at many London hospitals, because I've got friends working in various intensive care units in London, in Essex, in Kent, in various places. So what they had been telling is after August, they started having issues from September onwards. So they had been running continuously. Their capacity has been overburdened, and they had been working really hard. And many hospitals are really, they cannot actually really cope at the moment. That is actually the real truth. And some of them are actually running at 200%, at even 250%. And we have actually seen few patterns how the disease have been behaving. We actually predicted that winter can be tough, and it has been tough. And what in the first, when it started, we didn't know how we didn't know much about the virus, how we treat the virus, what we do about the medical management of it. And in the last few months, we have learned few more things. And initially, if you look at it, any patient who was having difficulty in breathing, if they are struggling for oxygen, we used to put a tube in the windpipe. I'm actually talking for the the laymen who are here, who may be finding it difficult to understand intubation. So instead of intubation, I'm using the word putting a tube in the windpipe. So if anybody was struggling to actually get the enough oxygen into their lungs or into their blood, we were actually putting a tube very early. But over the last few months, we have learned that putting the tube early may be not that good. So we have now changed our practice into actually using non-invasive steps. We use non-invasive ventilation. So we are using more and more non-invasive ventilation. We have actually got few numbers, and what we have seen is basically if you look at the hospitals, we have 200 patients in the hospital, but around 10 patients in the intensive care unit. So what that means is around 5 to 10% of the hospital patients require the intensive care unit. And out of that 5 to 10% of the patients, so a lot of them are actually looked after with a non-invasive ventilation. And what we have seen, I'm just talking about Milton Keynes. I think that is probably what we are seeing nationally. The mortality when you're actually having non-invasive ventilation. So you have got hospital patients, then we call as level 1. So if you're in a hospital bed, it is a level 1. Then from the level 1, if you require anything more than level 1 care, if you little bit more higher care, it is called a level 2 care. So that is when we use non-invasive ventilation. And in the non-invasive ventilation, what we have seen is basically the mortality is around 30 to 40%, the non-invasive ventilated patient. And when it goes to the invasively ventilated patient, what we are seeing at the moment, the mortality is almost like 80, touching 90%, and even going to 100% actually, above 80% intubated patients. And age-wise, what we are seeing, majority of the patients who are in the intensive care unit are above 60. It is not that we haven't got any young patients, but majority of the patients are above 60. And a lot of the patients have got comorbidities like hypertension, diabetes, and obesity. These three things are there, a lot of the patients. There are few young patients, quite a few, very young patients, anywhere from 20s, their 20s, sometimes even without any medical problems. And the other thing which we haven't actually found is that basically there is no way we can predict who is actually going to be sick. So that is another thing we have learned. And in the second peak, we do have few things, as you have learned over the national media. One of the things is dexamethasone, which is one of these cheap steroid, which we are using now. And that has definitely reduced the morbidity and mortality to some extent, but definitely it is not a cure. And I will stop there, and then I will actually, I will come back if you have got any questions or queries.
Thank you, Suresh. You've raised very important points there. I think you spoke about the capacity of the NHS, sort of reassured us, but still to say that it's not, we're not over all of this as yet. You've spoken about an important point. There's a question about morbidity and mortality in the UK. I think people are comparing it to India. We'll come back to that in a moment. And most importantly, you said we can't predict who will become severely unwell. There was a question to you from Satyan. And Satyan is asking your experience of using tocilizumab when compared to sarilumab in severe COVID pneumonia, ventilatory support, requiring ventilatory support.
So that's interesting. Actually, I was actually hoping that question might come. So at the moment, actually, we haven't got any. The trials are still going on. So I think it is too early to tell. Like the dexamethasone, which came the recovery trial, and the dexamethasone, we had a sort of a final sort of report which showed that dexamethasone is effective in some patients. Like that, early reports have come that tocilizumab is has shown some benefits, but we haven't actually got the real numbers. So I would say it's still early days. So maybe we have to wait some more time. It is shown some effect, yes, some benefits.
Satyan says that they've started to use it now.
Yeah, we are using. We are definitely using, but how far it is effective, it is definitely shown some benefits, but we haven't actually yet got it.
Sina has a question for you. What is the mortality rate for the new variant?
I think somebody, I think was it Pradeep actually touched upon that. Yes, it is more transmissible, but it hasn't actually, means what we see now is basically we have got a more transmissible variant. So you have got more cases. So when you have more cases, of course, there are more people who are hospitalized. And when you have more people hospitalized, then of course, there are more people in the intensive care unit and you have more death. But that doesn't translate into that variant is particularly more, doesn't produce more mortality. You've got more cases, so depending upon the number of cases, there is more proportion of people who are dying.
Thank you, Suresh.
Explain?
Yes, I think it does. So thank you, Suresh. I think you brought in again the pressures. A quick question from me, Suresh. Sorry, were you about to say something? Go ahead.
No, I was thinking. I'm sure because Gafur is here. He's working. He works in Apollo Hospital, and I'm actually really fascinated and amazed by the what do you call the statistics from India, because I was actually looking at all the graphs like a little earlier, comparing US, America, France, India. For the last few months, you have got various data per million. the infection rate, the mortality, the number of days in the hospital, all the statistics are available online. It's really amazing data from India. I don't know, so I will be really interested to see the data from India and how they are actually doing. I'm sure that India is doing much better than us, looking at all the numbers. You had a query actually.
No, my question was again about the human angle, which we might come back to later. Because
yes, there is quite a lot to actually talk about that.
Yes.
The psychological.
Perhaps we'll come back to it at the at the end, if you don't mind.
Definitely. Yeah.
Can I bring in the next speaker? Thank you, Suresh. Thank you so much for that. Can I bring in Pramod, please? So we have Pramod, who is, there we are. So Shadri Pramod, who is, so he's from Trivandrum. He's a consultant endocrinologist and a clinical lecturer, honorary clinical lecturer at the University of Bristol. He finished his postgraduate training in general medicine in 2000 and in endocrinology and diabetes in 2016. And he's a consultant in diabetes and endocrinology, general medicine since 2017. He gained experience in COVID by managing COVID in hospitals, so very practical issues there, keenly observing the epidemiological trends of the pandemic, mainly in the UK. Over to you, Pramod.
Thank you very much, Arunji, for that very kind introduction. And thank you to Kala and to Sumar Jatan for giving me this opportunity to share some of my thoughts, primarily as a clinician, and also as having gained some interest in some of the epidemiological trends in COVID. So as like everybody else who has talked here, I will start off with some of the experiences, what changed with COVID, and then I will talk something about the epidemiology of COVID. I am not an epidemiologist, and therefore I can't claim any authority on this. So do correct me if you think I have gone wrong. And I will also give you some of my perspectives as to what holds for the future. So coming back to my experiences, COVID became sort of a major issue in the UK in March, but we knew it was coming in January. We clearly knew that this was on in January, but we didn't know whether it will strike us. And then we had more and more indications from Wuhan. Things spread to Italy, Iran, and South Korea. And then we knew that this is real, we are going to get it. And then we got it in March. And I actually went to a conference in the first week in March in London and sat with a lot of my colleagues. And I can imagine we will never do this now. We were sat close together, chatting, no mask, no social distancing, in a closed room for a full day and came back home. This was all clinicians from England gathered in one place. You will never even imagine doing this now. So we can see how dramatically things have changed here. And I can remember that our WhatsApp chat in Kala was actually at its peak during that first wave. And we could see, everyone could see that the scenario was changing week on week. And you could anticipate it after a few weeks that hospitals are closing down. We are no longer encouraging routine admissions. And it became a very, very much a COVID focused care. And obviously we had taken lots of precautions at that time. And the first wave brought about a lot of changes, just like how Pratap and Pradeep mentioned about the fact that we changed over to telemedicine very quickly, something which we had never thought about doing as a change. Even when we had pressures from management to do so, We did that overnight, and we had to postpone some of the routine work, and then we got routine work going. COVID thankfully came down after the first wave, but now we have got this new challenge, which is to maintain a normal service hand in hand with an even worse second wave. So this is where we are at the moment. And that has posed a lot of challenges. Even in the first wave, we had a lot of frontline health workers having challenging mental health issues. And this is going to be something we have to anticipate, and hopefully we can provide each other support so that we can gain strength from each other to face the coming challenges. So even in spite of these challenges, there are a few things we can really be proud of. The first thing is how we have worked as a national unit, which is the NHS, and how we have brought about some groundbreaking treatments which has changed the natural course of the disease to some extent that we could save lives with very cheap medications. And if we didn't have these medications, the second wave would have been far worse. So we've got dexamethasone, and the way it changed practice was overnight on the basis of a press release. We would normally wait for trials to be published, and then we would discuss it, and then there's a gradual changeover. This was overnight. I even called some of my colleagues, including Rajiv Jayadevan from India, who is one of the former IMA secretaries in Ernakulam, who's a very prominent face in the media, and said, look, you have to start this tomorrow. You don't have to wait. So that was the kind of changes we had. And we have also got big epidemiological studies happening nationally, which is looking at antibodies in healthcare professionals on a national basis, which is already giving us information about how people who have been infected during the first wave have got equivalent protection to what a vaccine would give. So these are huge bits of information that we are sharing the world. So on that note, I would just like to share you some very basic epidemiological data and some simple reminders that everyone should continue following, because this is not the time for us to be complacent. So if I just try and see if I can share my slides.
You should be able to.
Okay. I hope you can see my slides.
Yes.
Okay. So I think it is important to just quickly have a perspective about how COVID-19 compares in terms of its transmissibility with other viruses. It is much less transmissible than a common cold, but much more than influenza, which is why one good thing that has happened with masking and social distancing and so on is that we don't have any influenza that is significant to be causing deaths currently. So we hardly see any influenza cases. We have Suresh Menon from Milton Keynes Intensive. I'm assuming he will agree with me on this. We are hardly seeing any influenza-related admissions to ITU. Thankfully, COVID-19 is not as bad as the SARS virus, but it is still way more serious in terms of death rates and long-term complications compared to influenza. And so we really need to take it seriously. Most studies which have looked at COVID-19 have shown that there is a higher death rate compared to influenza. Now, if you look at the calculations of deaths per infections, what is called as an infection fatality ratio, how many people die per 100 infections, we generally find that it is around 0.78 to 1.79% for high-income countries. It seems to be much lower for low-income countries, mainly because of the younger population there. And what we had somewhat hoped in terms of a reduction in infection fatality as time goes by, unfortunately does not seem to have happened yet, but hopefully with the treatments like dexamethasone and tocilizumab, and also the vaccines coming into place, we will start to turn the corner. Now, the infection fatality is not a constant factor. It's highly variable depending on the age and other medical comorbidities, as has been mentioned before, diabetes being one of the main ones, and also socioeconomic status. If you come from a poor background or aged between 30 and 40, your risk of dying is similar to somebody from a rich background or a higher socioeconomic status who is aged between 60 and 70. So this is something which needs to be addressed in the future. Some of the inequalities needs to be addressed so that we can bring about a global difference to COVID-19. Now, one of the questions that gets asked very often is, why do we have this strategy of a lockdown? Why do we need to have a lockdown? This is a bad strategy. Unfortunately, lockdown is not a strategy. It is something inevitable that happens because things have gone out of control. This is not something that was planned in advance. This is like somebody invading your country. You have got to defend. There is no choice. You can't go and hide in the bunker. You have to come out and fight. And that is what lockdown is all about. But what it does result is in a dramatic drop in infections and consequently reduction in hospital admissions and deaths. And what would really be a long-term effective strategy would be to do something between lockdowns, which is truly effective. We learn from what went wrong between lockdowns to prevent further lockdowns. We still have to practice wearing masks, physical distancing, and regular hand washing as usual. And obviously, vaccinations will hopefully make a difference in the future, but until we know for sure, we still have to continue these new normal practices. And just as Pradeep and other colleagues mentioned, we have to keep a constant surveillance on the infection rates and also on virus mutations, so that we can identify when things might be changing early enough so that we can act on it. And when you find that infection rates are going up after it has come down, we act early to break the chain. And this needs to be a constant process, and we can't have any room for complacency. Healthcare workers need priority because they have 9 times higher chance of getting severe COVID, and this has been conclusively proven in studies, and they need adequate personal protective infection and possibly vaccination priority as well. Monitoring international borders needs to continue. Very importantly, we need to maintain trust and have an open and timely communication so that people can engage early enough. There is always going to be the tendency when things start to get better that we bounce back to old normal. But I think we still have to persevere and keep adjusting to what we have come to know as the new normal condition. Now, the eventual strategy, as I see it, from COVID-19 will hopefully work with vaccines, better treatment, and social measures, but this has to be a global approach. The problem is global, the solution has to be global. Thank you.
Thank you, Paul. I think yours was a very useful presentation. I'm really sorry because we are sort of starting to get pressed for time, as always happens when we move on. There are lots of questions, but we'll come back to that in the general panel if you don't mind. Can I bring in the next speaker? Thank you so much for your very interesting, lots of points that you've raised about quick changes, the epidemiological data, the inequalities, the use of the lockdown, and measures that we need to take. Thank you, Pramod. do stay back and we'll bring you back. So I'd like to bring in the next speaker, who is Dr. Jose Matthews. Dr. Jose is he's a twin specialist. So he has a background in pediatrics, and he's currently a GP, 30 years in medicine, 14 of them as a GP. And so he's a native of Kodamangalam, as all of us, homegrown, studied in Kunur, and he's a St. John's Medical College alumnus. No, sorry.
No, it's okay. Don't worry. Carry on. I'm not, but you carry on. Don't worry.
Okay, you can tell us where you're from.
Yeah.
Okay. So you currently work in Reading, and you're involved with the COVID immunization programs. So over to you, Jose, and you can correct me.
Yeah, no problem. Don't worry. First of all, let me share my screen. Can you all see?
Yes.
Yes.
Okay. Initially, thank you, everybody. And I hope everybody is keeping well. Nobody has got any symptoms or is worried or anything like that way. The general news which is going around makes us all worried. We are frightened. We feel there is no hope. We feel that everything is going to get on top of us. So I've just put a little slide up here which shows what the various pandemics and epidemics and all that have caused, how many people have been involved, and all those kind of things. So if you see, we are coming closer to the top, but not too bad. And in this, I have not included the flu pandemic in the early 1900s. So let me just take you the step forward. First, it started in Wuhan, as we all agree. Currently in the UK, this is the situation of the number of infections and the deaths. This is correct as of yesterday. Today's ones, I'm not sure. And because it's a weekend, I'm not entirely sure whether they will have correct numbers for it. So that is the situation. The worldwide situation is slightly different. And if you look at that, I've put the Spanish flu and also the current SARS-CoV-2 one together. Spanish flu infected 500 million. Currently, ours stands at 93 million. Between 17 to 50 million people died in the Spanish flu. And it's around 2 million plus a little bit more now. So looking at that, there is some cheer in as much as that, yes, we have a pandemic. We are only 12 months into the pandemic, usually lasts between 18 and 24 months. And we are still a way off from the total Spanish one. Advances in medicine, advances in care, advances in social delivery, advances in various other fields may account to a certain degree for that improvement. Then also the characteristics of each individual pathogen also plays a role in that. Then the next one is signs and symptoms. Everybody has looked at these. Most of us know them by heart. All that I want you to do is to take that last link to heart. This puts you through to a website, which is what we inform our patients, and tells you what to do when you have certain set of symptoms, where to seek help also. Working practices. This is the most important change in general practice. You have always in general practice, mainly, I mean, not always, mainly, you have had a telephone call to the receptionist. They book you into the surgery. You come and see the doctor. The doctor gives you a little bit of time, whether it is 2 minutes, 5 minutes, 10, 15, 20, depending on what the situation is. And then you, to a greater or lesser extent, feel, okay, right, I've been sorted, and I go home. Or you come away from the consultation saying, oh my God, that was disastrous. I don't want to go back there. Whatever it is, you have had that personal touch. And that personal touch gave you a lot of satisfaction. At least that's what I know from my patients' feedback. Now that has changed completely. You are doing it totally, 80 to 85% is on the phone, with or without video, and less than 15% is through face-to-face assessments. most of the GPs will do an initial triage, and then based on the triage, if they are safe and they need to see the patient, they'll bring them in, and then we have a look at it. This provides a little bit of risk to the doctor and the patient. The difficulty with this is that some visual cues, some things which we found by looking at the patient, the way they behave, the way they interact with us, all those things have disappeared. We have to assess them remotely. How do I sort of assess a patient who has got a breathing difficulty on the phone? Next to impossible. But we have learned new techniques. Luckily or fortunately for us, we had a previous system whereby we had a part of our sessions done solely by telephone triage. So we were in a better place to sort of change over from one system to another system. There is mass trauma out there. Everybody's frightened. I've got it, you may get it. Third party may say, well, I may get it in the future. I've lost my job. There's no place to go, no money. I do not know how I'm going to eat. I don't know where I'm going to get my house from. Hundreds of problems like this way. And this trauma is amplified by the fact that we do not have answers. The epidemic is actually just progressing. New things are coming in. New ideas are being shared. And the amount of information which is coming through on a weekly basis, on a daily basis from multiple sources is massive. We have to disseminate that. First of all, we have to take it in. We have to learn. We have to find out what's going on. Then we have to disseminate it to other people. This is difficult, but somehow we manage to be coping in a certain extent. There is ethnic and social and cultural variabilities. The same thing that you feel is okay, another person may not feel is okay. There's resistance from certain groups towards vaccination. There is resistance from certain groups to believe that this is a problem, that there is a significant amount of illness out there or infectivity going on. So all these things are to be taken into consideration in a general setup. When you go down the specialist setup, then you deal with that particular issue and that particular system or that particular problem. We have still got a long way to go in this. Language barriers. I speak a language, you speak something else. I tell you one thing, you understand something else, you learn something else, or you take in a different bit of information. All these things are difficult. Then comes managing uncertainty. As a generalist, you always will have this situation. Do I test him? Do I keep him for observation? Do I send him to the hospital? Do I just tell him to go home and see how it goes? But there are some techniques which we learn to sort of minimize this uncertainty and also to sort of safety net around it. Then anxiety. That's the biggest problem that I've noticed in this pandemic. Everybody that we talk to is anxious. They're either anxious about the illness or they're anxious about the outcomes or they're actually anxious what could happen. And it's difficult to manage this. We try some techniques. We also signpost them to some service providers. Maybe it's helpful. Let's see how it progresses. We have not been able to do our routine preventive and therapeutic work. Like, for example, monitoring diabetics. There are less number of people coming in. They know they are at higher risk, so they access the service less. Even if you ask them to come in for a blood test, they'll say, well, you know what, I'm coming to a GP surgery. What if I catch the illness? All these challenges are there. Then mental health. Mental health and more importantly, safeguarding. We have got a large population there that have slipped off the radar because we have got no way to contact them. Previously, the school system was there. The nursery system was there. The health visitor service was there. GPs were there. All of us put together, social work, social services, all of them put together would keep an eye on everybody. And when things started to go wrong, some people would step in and say, listen, this is not right. Let's have a look at you. Now, because of this block, we don't seem to be able to get to that same level. So that is also a big issue. Then we have got the workforce. As a doctor, like one of my colleagues said, 9 times more likely. If you have got 9 times more likely to be seriously unwell And if you become unwell, you're going to definitely take the time off. While you're off, there is nobody else to replace you in a small surgery. So what do? You have to reduce your service, or you have to get into collaborative working. We had started collaborative working. This seems to be going along quite well at the moment, and hopefully it will continue that way. With every challenge comes opportunities. So the challenges are, how do we keep the workforce happy and well so that we are motivated to effectively manage our clients? Some simple things, words of encouragement. The government said, these NHS heroes makes good thing for us. It says, oh, you're valued, you got thing. Clapping, that was also something valuable. All these things put forward a picture out there that you are needed, you are part of the system, and you're welcome with everything. By the way, my family is laughing at me. Then comes the challenges of media and reporting in the connected world. Everything, say for example, those 2 people who had developed the allergic reaction to the vaccine. Immediately it was in the news. Everybody was saying, oh, no, I don't want to have this vaccine. But people say they're getting allergy and all that. Unless it's a balanced opinion, it is difficult for the lay public to say, well, that's reasoned out. This is the proper way to do it. 2 out of 44,000 at that time would be nothing. It would be very low incidence of any kind of complication. Whereas if you had it 2 out of 20, that would be very high. So you have to take it in balance. Then changing the way we work, remote working, longer consultations, online work, all these things are going to come in. I think in the future, it's very unlikely that we're going to go back to the old system where every patient who has a phone call comes in and sees the doctor. It's always going to be a combination of some things which are going to be remotely done and some things which are going to be done face-to-face. For example, if you've got a lump or a swelling somewhere, you cannot do that on the computer system. You have to come in for someone to feel it, touch it, see it, say that this is the likelihood of that, and then give you an answer as to what is the best way forward. Whereas if it is something that you need a sick note, yeah, I can speak to you on the phone. I can have a video consultation, make a small assessment and say, okay, fine, your sick note is provided. I'll send it to you electronically. Then the NHS at some stage has to restart because we have got all these things which are commonly seen and conditions which could have waited are waiting. We have got to catch up with that. We have to go back and start to take care of all of this. Knee replacements, hip replacements, seeing you for your deviated nasal septum, nosebleeds, all these things. So we need to get all these things restarted. These people who have not been accessing healthcare have delayed their presentation. So they will now start to come in slowly as the pandemic starts to regress. Then you will have a little bit of a catching up to do. How many investigations, what treatments, we have to try to make these things go forward. The long-term conditions that we have not intervened with or have put on hold at the moment have got to be restarted, bring them back, make them all sort of managed or well managed as possible. Then the rise in mental illness. Most important now is the delivery of the vaccination. I didn't mean to write flu vaccine. I meant to write the COVID vaccine. This is a very large effort by the NHS. You have got to take care of everybody above the age of 18. Everybody that wants it or needs it should get it. Till now, we have had annual flu vaccines. And the annual flu vaccines would be above the age of 65 or those who have got other medical conditions. Plus, you need not have to observe them post-vaccine. Whereas this one, you need to observe them for 15 minutes. The government may change the guidelines, but currently, this is the situation. So it's going to be a massive thing. We are doing pretty well at the moment. I hope it stays that way. And I'm sure with the NHS being what it is, it will manage. Protect and prevent. What can you do? Wear a mask over your nose and mouth, whether it is going out for coffee, to buy essentials, or whether you're in your office, put down a mask which covers your nose and mouth. Continue. That is You have to use the 2-meter rule between yourself and others. The mask is not a substitute for this. You put the mask and the 2 meters. Wash your hands as often as you can. Soap and water for at least 20 seconds. Or if you want to use a disinfectant, you can try that also. Different guidelines. Avoid touching your eyes, nose, and mouth with unwashed hands. Always cover your mouth and nose with tissue when you're going to cough or sneeze, and then bin it. If you don't have a tissue, use the inside of your elbow and do not spit. This is particularly so in some cultures. Throw used thing. Be alert for symptoms. If you have the symptoms that are described, please isolate and test so that you don't spread it. Testing, these are the various testings. Currently, the RT-PCR is the one that we are using. Lateral flow test is also coming in now. The relative degree of accuracy is also given there. Other tests are also being trialed. As they come in service, we'll start using them. Then comes the business of what happens after COVID. After COVID, there is long COVID or post-COVID syndrome. Different people have sort of described it as symptoms lasting for more than 4 weeks, which I said recently, and most other people say more than 12 weeks of the symptoms. And the symptoms I've listed below. This is going to be a big drive now. If people remember in Kerala, there was one chikungunya. And after that, you found a lot of people had persistent joint pains, aches. They couldn't walk properly. They were bent over for quite some time after that. Similar to that, we have got this sort of symptom where a lot of people are getting symptoms which are quite vague, affecting many systems or different systems. And the NHS has recognized this, and they have started long COVID clinics where once the diagnosis is made by the general practitioner, they then send you to these clinics where they get a multidisciplinary team which helps manage all these symptoms and hopefully makes things better for you. We are still stuck with the knowledge about the condition. It is evolving. As time goes by, we'll better understand it, and then maybe we'll be able to manage it better. Finally, the vaccine. There are 3 candidates licensed in the UK, and those are the 3. Moderna is the last one. I didn't put that as a separate one. Currently, only the Pfizer and the Oxford ones are the ones that are being used. Moderna will come in soon. You need to have 2 doses between 4 and 12 weeks. The government has decided that we will do the 12-weekly routine. And the reason for this is to make sure that more number of people get the first one so that there is some protection in the community as trying to get a small number of people vaccinated twice so as to get full protection. And finally, the order of vaccine. We are currently running in that anyone 75 and over category in most places. I'm not saying everywhere, in most places. So we have been vaccinating now for roughly around about coming up to 3 weeks. So we have already reached the 75 and over. So very soon we should be able to start getting into the lower categories and thereby the whole population covered. And that's about all I need to say about it. Any questions?
Thank you, Jose. Thank you so much for that very comprehensive presentation. And I think what you brought to the forefront is everybody's been told that the NHS is struggling. I think you've brought to life one of the pillars of the NHS, which is the general practice. And sorry, Jose, we might need to move on because
Jose, I don't know. You are not. It is me who gave the wrong information. Jose, what I said, St. John's.
Ramaya.
Okay. That's all. The others are correct.
All the others are correct.
I don't. It's not. It's me, Jose. Okay.
Okay. No problem. Cheers.
Thank you, Veda, for saving my skin.
Yeah.
I think we should, Jose, thank you so much. So we'll move on to the next presenter. And I would like to thank the next presenter, who's Dr. Abdul Ghafoor, who has been patiently waiting for us in India. It's 10:30 there, Abdul Ghafoor. Thank you so much for waiting. So Dr. Ghafoor is a consultant and adjunct professor in infectious disease in the Apollo hospitals in Chennai. He's a coordinator of the Chennai Declaration, which is the initiative to tackle the superbug crisis. He's active in academic, political, and policy aspects of infectious disease, and he's a jury member of the Longitude Prize. So over to you, Dr. Gafoor. Sorry, we can't hear you. No.
I think you may need to change your audio device to your AirPods on Zoom, where you have the mute button. You can go there and choose your system. The microphone and speaker needs to be your AirPods. No, not audible. No, not yet. Yeah, I think you're on.
Hello.
Yes. On.
Sara kekamo?
Yes, you're ready to go.
Welcome.
We can hear you.
I think he cannot hear us.
I can, with the AirPod, I can hear you.
Yeah.
Okay, yeah, we can hear you.
Oh, okay, fine, great. Okay. Namaskaram. In the beginning of the pandemic, many Western epidemiologists and organizations predicted India is going to be the COVID graveyard of the world. No such apocalyptic event happened. India is still alive and kicking. At a global level, more than 100 million people are tested positive for COVID and more than 2 million died. In India, 10 million people are tested positive and 150,000 are dead so far. So the case fatality rate in India is 1.5, but at the global level, it's around 3. So the CFR in India is half of that of at the global values. So why India is having a comparatively lower case fatality rate? Is it because India is underreporting? No. There may be some level of underreporting. The value may not be perfect, but definitely we are not underreporting to a great extent. It's really the CFR in India is low. One of the main reasons could be the age. Most of the people who got COVID in India are young, and of course, the low mortality. It could be because of the cross immunity, because of constant exposure to other respiratory viruses and other viruses. We don't know the exact reason. These are the hypotheses we give as an explanation for the low CFR. At a very early stage of the pandemic, India went into lockdown, very, very early stage. And that really affected the migrant population of the country. But overall, the lockdown helped India, at least in the early part, because we could increase the setup of the COVID management system in the country. Government system predominantly managed the COVID. Later, the private system participated. So you all know our problems with our government setup. I have to say our government setup rose up to the occasion and managed the COVID reasonably well. And now India has initiated the world's largest COVID vaccination campaign, vaccinating 300 million people. And 300 million people means that's the whole population of America. In UK, you have 66 million. And India is going to vaccinate 300 million Indians in 6 months. That's a massive campaign. As we know, we have enormous experience from our National Immunization Program, our UIP. And we have enormous experience of conducting national elections. and then we have the public participation. We are going to utilize all this experience to make the national COVID vaccination program a big success. Just now I was listening on the TV, I watched on the TV, I was listening the statement from Boris Johnson. He was congratulating the Prime Minister, and Boris Johnson told that India is going to be the COVID vaccine pharmacy for the world. More than 50% of the COVID vaccine for the world will be manufactured by India. And we have, when we initiated the campaign yesterday, we are utilizing the Oxford University vaccine, the Covishield, and the Bharat Biotech India-made vaccine, Covaxin. We are going to utilize both these vaccines for our program. In 6 months, India will be completing the vaccination of 300 million public. And that's going to be a huge success. Let's wait and see. Thank you.
Oh,
Dr. Abdul, we did not introduce you, who you are. Slide on short time. Vitamin D part on the ഒന്ന് പറയും നാട്ടിലുള്ളവർക്ക് വിറ്റമിൻ ഡി കിട്ടിയിട്ടാണ് അതോട് വിറ്റമിൻ ഡിക്ക് പ്രൊട്ടക്ഷൻ കിട്ടുന്നൊക്കെ പറയുന്നല്ലോ അതിനെപ്പറ്റി എന്താ ഒന്ന് പറയാമോ?
I never prescribe vitamin D to any of my patients. I treated hundreds of COVID patients. I never prescribe vitamin D to any one of them. Some of the state policies have included vitamin D, but we don't use that routinely. And I don't prescribe vitamin D to anybody without vitamin D deficiency. So I don't think it's because of vitamin D, and that's not a part of our national program anyway.
There is one question from America, one of caller, asking link with the fertility and the vaccine.
Fine, okay. That's I don't know it's a conspiracy theory or not, but there is some hypothesis. Because if you look at the structure of the spike protein receptor binding domain, some of the scientists hypothesize that there is some cross reactivity with the spike protein, and there is an antigen in the placenta called as syncytin-1, and you can have a cross reacting antibody after vaccination and transient infertility. But then that's possible with COVID infection as well. If there is a cross reactivity with the spike protein of the vaccine, then of course that is possible with natural infection. It's not proven. It's just a hypothesis. You know, it's not a proven. I don't think we should be worried about infertility through COVID vaccination anyway.
That's very good. Why? I don't know if there is another few more questions. Perhaps you could. Otherwise, I will be asking at the end. If time permits, I will ask.
There were a few questions from YouTube. There was one specifically, I think, for you, Dr. Gafoor. I think Tangur on YouTube was asking, what's the ethics of allowing Covaxin to be administered while the phase 3 trial is still in progress?
A very interesting question. See, we are initiating the world's largest COVID vaccination program. We have Covaxin, we have Covishield, but they may not be sufficient to vaccinate our 300 million in such a short span. So we are utilizing Covaxin, which is a kill whole virion, whole virus vaccine. And India has got enormous experience of developing and manufacturing killed vaccines. Kill vaccines are comparatively easier to make. We have got extensive experience, and kill vaccines are relatively safe. The Bharat Biotech has got phase 1 data, phase 2 data, which is immunogenic and safe. The phase 3 will be completed in a month's time. In my opinion, Indian government should have introduced this vaccine 2 months ago when the company finished phase 2, where we had larger number of COVID. In fact, I wrote to Health Ministry suggesting, why can't you introduce this vaccine after phase 2? Because we have a large number of COVID in our country. It's a kill vaccine. Why are we waiting for phase 3? Let me ask a question. Let's look at the Covishield of Oxford University. If you look at the phase 3 trial data, only less than 10% of the phase 3 population is more than 55. Who are you vaccinating? Elderly first. Is that Oxford University phase 3 data sufficient enough to vaccinate elderly? the safety and efficacy of the Oxford vaccine in elderly is not proven. If you look at the Lancet publication of the Covishield, it's very clear. The data is not sufficient to calculate the safety and efficacy in elderly. Are we excluding the elderly in vaccination in UK or India or anywhere in the world for the Covishield? Are we waiting for large-scale phase 3 data of the elderly? We are not. So if you look at the deficiencies of vaccine trial, you can find deficiency with almost every trial. Look at the Pfizer vaccine data, phase 3. If you look at the adjudication committee of Pfizer, its adjudication was done by 3 Pfizer employees. Is that ethical? I don't think so. So if you look at the minor deficiencies of vaccine trial, we can find trial with any vaccine. Everybody is bending the rule. Yes, nobody is cutting corners, but some level of bending the rule has happened in all the vaccine trials because we are facing a global pandemic. We cannot be perfect. We have to be safe. What is more important is safety, and safety is proven after phase 2. So I don't think what Indian government, I support what Indian government did. I even actively supported and still support what Indian government did. It's a good strategy. We are facing a pandemic and it's a kill vaccine. It's safe. We don't know how efficacious it is because we need phase 3. It is immunogenic. More than 90% individuals in phase 2 had neutralization antibody. So it's really immunogenic, which is a surrogate marker for efficacy. Of course, we will have the phase 3 in a month's time, most likely, and we'll get the exact value. I don't think we have to wait for completion of the phase 3. Many people have criticized, but my argument is there is deficiency in every COVID vaccine trial. None of the COVID vaccine trials are perfect. You can find fault with any vaccine. But let's not try to find faults. Let's look positive and let's make the COVID vaccination a great success, and so that we can end the pandemic.
Thank you, Dr. Gopal. I think you've been quite vocal in that. I think there are a few other questions, but perhaps those questions could be asked to the general panel if we have a little bit of time at the end. I can bring in other people as well. But thank you for your patience. It's quite late in the night. If you can stay back with us, it will be wonderful. Dr. Ghafoor, so if you could stay back with us.
Sure.
Okay, if it's not too late.
That's very good. Okay.
Thank you, Dr. Ghafoor. I'd like to bring in the next panelist, who's Mr. Ramachandran.
Mr. Ramachandran needs to turn on his video so I can add him. I've just asked him. Oh yeah, he's on now. There you go. He's here.
Oh, Namaskaram. Namaskaram, Ramendran.
Unmute ചെയ്യണം.
Okay. ഒന്ന് unmute ചെയ്യാമോ രാമചന്ദ്രൻ? Unmute ചെയ്യാമോ? Okay.
Okay, now it's okay?
അപ്പോൾ ഞാൻ നേരത്തെ പറഞ്ഞ സമയം ശരിയായിരുന്നില്ല.
No, that's fine. Okay, first of all, let me tell you good evening to all of you. It was a wonderful team, and everybody's presentation has been great. And the last speaker, Dr. Abdul Ghafoor, has given a really very, very informative information about the clinical trials, etc., which has been very eye-opening. As a matter of fact, my role, after having heard all these speakers, is very limited. I don't know whether, because I'm a manufacturer of the general medicines, which is for which I don't have any kind of experience on the vaccines, rather than whatever we are hearing from the others and whatever the information we are collecting. So therefore, I may not be able to contribute anything substantial to this team about the vaccines and their applications and their clinical trial applications, etc. However, whatever I can know about the general medicines which are used, probably a supplementary treatment in case you have a fever, you can take paracetamol, or you have any other thing. They are our core area. We have manufacturing units in UK. We have been manufacturing continuously without any interruption so far, even though we were passing through a very bad time. I would rather look forward to receive your questions if you have anything to add into that these things, because the team what has been presented, I may not be fitting into that there in this discussion. So that is what I wanted to say in relation to the vaccines.
ഇതിനകത്ത് ഇപ്പോൾ ഉള്ള ഒരു സംശയം, ഇവിടുത്തെ വർക്കിംഗ് കണ്ടീഷൻസും ഇന്ററപ്ഷൻസും ഇത് കറണ്ട് സ്റ്റേറ്റ് ഓഫ് ഇൻ UK എങ്ങനെയാണെന്നുള്ളത് പറഞ്ഞാൽ കൊള്ളാമെന്ന് തോന്നുന്നു.
Yes, we are definitely going through a very tough time. Absenteeism is one of the major concern, because a lot of people who are workmen coming to the workplace are fearful about the possible COVID this thing, and because of that they fear and they do not come regularly, and so we find a large number of absenteeism, which is resulting in poor productivity. However, till this date, we have been able to supply medicines to the market and also to the NHS hospitals so far, irrespective of various constraints. We also face a lot of issues related to getting the importation of the raw materials from countries like India and China. There has been an abnormal delay in such deliveries, and this is also resulting our productivity. However, so far we have been able to manage. I don't know how far we will be able to continue like this, so which is yet to come, yet to watch and see.
Okay, financially
it's a lot of concern, because you know that when you don't have, you utilize your capacity in full swing, or you don't produce the required quantities, you are not able to meet the cost. And as it is, the cost of labor cost in UK is 10 times more than India or any other countries. So therefore, we already have a large overheads. So a less productivity keep us down in relation to our revenue as well.
One more question, Ramendran. Whatever medicine you are producing at the moment in your factories, because if it is not running to the full swing, if it is so, is it going to affect the supply of medications to the NHS?
So far, we have been able to manage the supplies to the hospitals, whatever the products we are contracted, also to the marketplace. However, there is a lot of uncertainty on a continuity of full swing supply in future. It all depends on the availability of the raw materials and also the efficiency of running the plant, because this is only can decide. If we don't have the people to work, then we cannot produce it. And then we are also to take a lot of precautions in the manufacturing area to protect the COVID-19 effective this thing infections. So which is why we have to be very careful, and I won't be able to make a 100% statement today that 100% supplies will be given. But however, we are trying our best, because this is our requirements, and we are also very constantly in touch with the Department of Health here. And wherever there is a shortage, we are definitely opening up those areas into priority and trying to supply, so that there is no interruption to supply to the medicines in the market, until the patient is getting the medicine what is required.
Thank you. Arun, over to you, Arun.
General panel questions. Okay. So thank you, Mr. Ravindran.
If you're able to stay, please do. Otherwise,
please.
Okay.
I request to everybody who has been participated, and thank you for inviting me. And it's a great pleasure watching this TV, which is this program. Very, very well appreciated.
Thank you.
Thank you. So over to you, Veda.
Okay, we are reached, even though we are a little bit late, we are having the last speaker within the panel, but there are more questions and answers to come. But I have to introduce our moderator, the anchor, Dr. Arun Kishore, as I said, a consultant psychiatrist, trained as a psychiatrist in 1998, and has been working as a medical teacher till 2006, and moved to UK, and now working with Practitioner Health, which is an organization supporting doctors in distress. So his interest is in psychotherapy, individually as in groups, and training to be he's planning to training to be a group analyst, and above all, he is a good interest in poetry, theater, has been performing a lot of play in our Kala annuals, and a good gardener and a bird watching person. Let us see what comes from his sharp eyes. Let us listen to Aaron. Okay.
Thank you, Suveta, for that introduction. I'll try and keep mine brief predominantly because of lack of time. And I think people have touched upon the issue of mental health in COVID. So I'd like to talk about this from two aspects. One is the issue of people who have mental health problems and how COVID affects them, and the other from the angle of people who do not have mental health problems and how COVID affects them and their mental health. Now, I must say that I am a general adult psychiatrist, and I'm fortunate to have my friend and colleague, Dr. Sina, who's an expert in child and adolescent mental health, and I'm sure Sina can supplement whatever I have to say amongst children. Thank you, Sina. So I'll invite your thoughts here as well. So from my side, Sina, my understanding is that initially, that when the pandemic started, there was a sudden quietness in mental health, almost as though our hospitals.
Sorry, what did you say, Arun Cheta?
When the pandemic started, it was almost as though we didn't have any patients in hospital. Most of them got discharged and left, and it seemed a very quiet phase. But it seems in the second phase that there's been an increase in mental health. I see so many patients. I see doctors in distress. I see the effects not just from the pandemic on mental health, but on the pandemic on normal mental health. And my worry is about how this might convert people who are normal in mental health into people having a mental disorder. What are your thoughts on that, Sina?
I can talk about myself. When it first started, I went through an existential crisis, and I was in a hurry to finish my bucket list. I have a bucket list of things I want to do before I die. So I started seeing that death has come closer and started working on my bucket list and became more focused. Talking to other people in my family, the anxiety has risen through the roof. Even normal people, like you said, not with any mental illness. In people with mental illness, we know that stress can cause relapses of people who are in remission. It can cause exacerbation of an already existing illness, and it can even trigger an illness which is not there. So it can trigger, it can exacerbate or cause relapses. So stress is bad. We know that in mental health. So in child psychiatry, I can talk from personal experience. I don't have any data, but in personal experience, the school refusers, the ones who did not want to school, initially went through a happy phase because they didn't have to go to school. But all the other illnesses, OCD, anxiety, schizophrenia, psychosis, all those rose up. And even abuse, because of being in a pressure cooker environment, abuse went up. I don't have statistics or data, but it went up. I don't know whether I've answered your question. And in normal people, I can only talk from experience of people who have been leaning on me and making phone calls about everything, about anxiety, about COVID, about future, lots of issues, planning the future, making a will, all sorts of things.
Absolutely, Sina, and I couldn't agree more about all of that. You've spoken about those special populations, and you've spoken about children, and I'm wondering, I work with a lot of people who are in the medical profession. I speak to nurses and I speak to doctors and I speak to other workers. I can see that most of them are under stress. So I run groups for people in India, doctors, and I am associated with the Practitioner Health Program, which again caters to doctors who are in distress. I work with the Doctors for Distress program in India, and the IMA has a program for that. What I see is that a lot of them are under stress. I have doctors come back to me to say, I wish I were not a doctor at this point of time. And it's terrible to hear somebody say that. I have nurses come back to say, I wish I were not a doctor. I wish I were not a nurse at this point of time. They're tired and they're quite exhausted.
Not just that, there's also survivor guilt about having survived, and it is too close to them, and the feeling of helplessness of not having saved them. So that is there. Plus trauma symptoms. Sometimes they are like a hamster on the wheel. They keep going, they keep going, they keep going. They don't have time to process what's happening. And it's later on that the whole trauma sinks in, and then they let go or they crumble. So initially it's a phase where they keep going and keep going, not having time to process what's happening. And then it's when they stop to think that the trauma hits them. There will be a lot of PTSD. It is expected, sort of PTSD sort of symptoms in any healthcare professional, ambulance drivers, not just medics and nurses. And death has become an alienating experience. For the people who die, death has become an alienating experience of not being able to say goodbyes. For the relatives, it's become there's no sense of closure. So the grief reaction is prolonged because there's no sense of a closure of saying goodbyes to their near and dear ones. So even death has become a very, very complicated issue. We have seen children who've lost grandparents or whoever, for whom they've not been able to attend a proper funeral because of the limited numbers and COVID protocol and whatnot.
It has, and again, I think for some people in color, we have our loved ones back in Kerala, back in India, and that distance and the problems that brings about. But quickly, Sina, before we sort of bring this to a close, what sort of warning signs could people look for? When your mental health is sort of sitting somewhere on the fence, when it's getting a little dicey.
Anything out of the ordinary, wouldn't it be? Arun Chetan, you tell me. Anything out of the ordinary, anything which is unlike the person's behavior. I don't have anything specific to say. If you feel that the person is behaving out of character, sleep getting affected, appetite getting affected, behavioral changes, moodiness, withdrawal, whatever. There's nothing specific, but anything out of the ordinary or out of character for the person.
Absolutely, Sina. So those 4 points came to my mind. One is, if you feel that you're becoming inflexible. Now, flexibility is so essential for us. Flexibility of thought, flexibility of emotions, that's essential for us. But if you find yourself becoming inflexible, then do think that you're under stress. If you find that your sleep or your energy levels are going out of kilter, I'd say watch your body. If your body is out of sync, and that means that there's something wrong.
Mm-hmm.
Watch your emotions. Either not having any emotions could be a problem, having too much of them, fluctuating emotions could be a problem.
Yeah.
Watch your thoughts. If you find yourself becoming inflexible, having predominantly negative thoughts, seek help.
Yeah, you summed it up well.
The final thing is about, now, one peculiar phenomenon I found is everybody's working from home. It means that families are thrown together. It also means that you see your spouse 24/7. And I'd like to sort of, and I'd like to look to you, what do you do in such a situation when you're thrown together? For me, one point that came up was about don't sweat the small things. Which means, small things are important in our lives, and there are small issues that might crop up. Forget, forgive, and move on. Don't sweat them. What else would you say?
I think there is no one glove fits all. There is no universal formula for the whole 6 billion population. I think it can go both ways. For some people, when the lockdown started, it was amazing because they were getting family time, and they found it a blessing in disguise. But for people whose relationships were already fragmented, it went worse. people whose relationships had undercurrents of unhealthy, dysfunctional relationships, it went worse. So it can go anyway, depending on the kind of relationship and the people involved. But what I'm saying is, I agree with you. Don't sweat the small sprouts. Is that what you said?
Yes, absolutely. Absolutely.
Yeah.
Thank you, Sina. I think we've taken on a huge thing. I don't think we can cover the entire gamut of
No,
mental
it's
health.
not possible. Yeah.
But thank you for being here with me. It was reassuring.
Thanks, Arunjeta.
So, Greta, over to you.
Okay. So we have time is running out almost, isn't it? It is 17:35. So Sita Devamur was about to come and say something about the community, and Ravi Shekhar as well, what is happening in the NHS, but we had a good discussion. So I think shall we conclude now, because it is 5:36 now. Shall we do that? Will you agree with that?
I think so, Sugat, and I'd suggest that most people take a look at our chat. There's a wonderful amount of chat going on between people.
Yes.
We're sorry that we've not been able to answer or ask all the questions.
Yeah. But, after this one, we are having say KJ Yeshdas 81, Dr. Yeshdas 81st birthday. The group is having some celebration with singing the good songs. People can those who are happy to join, please continue. We can continue with the questions and chat, but officially let us close it now, and then we will do it. But one thing I have to say, thank you to all the participants, YouTube participants, and friends, family members who are all watching now. A big thank you. I salute you for your support. And Kala is very, very happy that we could do this much effort in bringing the COVID and beyond. And all speakers, and I'm not going through the names because it has been done. And thank you, Arun, for coordinating and anchoring. And I have to say special thanks to Sham from Pittsburgh, because he's ഷ്യാമിനെ ഇപ്പോൾ ഉറങ്ങേണ്ട സമയമൊന്നും ആയിട്ടില്ല, പക്ഷേ നമ്മുടെ അബ്ദുളിനാണ് ഉറക്കത്തിന്റെ 11:30 മണിയായി. പാപം അബ്ദുള്ള, എന്നോട് ക്ഷമിക്കണം കേട്ടോ. ഉണ്ടെങ്കിൽ നമുക്ക്
special thanks to Suguchetan. You are the one contacted me for this, and it's a great pleasure to be here.
അതെ, എനിക്ക് തോന്നുന്നത് യുവർ ബ്രദർ ഈസ് വാച്ചിങ് തോന്നുന്നു കേട്ടോ. ജീവൻ ഇൻ പാരീസ്, isn't he? He's in Paris, isn't he?
Traveling somewhere. I don't know where he is going. I don't know whether he's watching.
എന്തായാലും അബ്ദുളിന്റെ വേറൊരു കാര്യം, അബ്ദുൾ, യു കുഡ് സ്പീക്ക് ടു യുവർ ടീച്ചർ. Am I right, Abdul? Are you there? Okay. Yeah, I don't know. Yeah, I think he has gone. Anyway, Abdul, are you happy to speak to your teacher? I don't. Come here. Come here. Okay. Thank you very much. എല്ലാ പാർട്ടിസിപ്പൻസും നന്ദി പറയുന്നു. പിന്നെ ഇതെല്ലാം അറേഞ്ച് ചെയ്തതിനും പിന്നെ ഇതിന്റെ പിന്നിൽ പ്രവർത്തിച്ച നമ്മുടെ ആർട്ട് സെക്രട്ടറി ശ്രീറാമിനും എല്ലാവർക്കും ഒന്നുകൂടെ നന്ദി പറയുന്നു.
ജീവൻ is online.
ജീവൻ, നമസ്കാരം ജീവൻ. അനിയൻ എത്തിയിട്ടുണ്ട്, നന്നായിട്ട് പറഞ്ഞു കേട്ടോ. ഓക്കെ.
ഞാൻ കേട്ടിരുന്നു, ഞാൻ കേട്ടോണ്ടിരിക്കുന്നത്. ഞാൻ ട്രെയിനിലാണ്, അതുകൊണ്ടാണ് ഞാൻ വീഡിയോ ഒന്ന് ഓൺ ചെയ്തത്. ഞാൻ
ഓക്കെ ജീവൻ. നന്നായി. നമുക്കൊരു കാര്യം ചെയ്യാം. ഇപ്പൊ എല്ലാവരും നന്ദി പറഞ്ഞ കൂട്ടത്തിൽ നമുക്കിനി നമ്മുടെ കലയുടെ പ്രസിഡന്റും സെക്രട്ടറിയും വിളിച്ചിട്ട് എന്തെങ്കിലും പറയാനുണ്ടോ എന്ന് ചോദിച്ചിട്ട് അവരുടെ വകയായിട്ട് ഒരു കലയുടെ ഒഫീഷ്യൽ ആയിട്ടൊരു താങ്ക് യു പറയണമെന്നാണ് എൻ്റെ അഭിപ്രായം. Let's invite Gopal if he's around here. I think
Gopalji join ചെയ്തിട്ടില്ല.
Okay. Mani, are you here?
Yes, I think he is here.
Mani, can you join? Okay. Your comments and thank you for words to all of the participants and everyone. Mani, which is I request you to do it.
Okay.
ചേട്ടാ ഞാൻ ആൾറെഡി എന്റെ കമന്റ് ഇട്ട് കഴിഞ്ഞു ചാറ്റ് ബോക്സിൽ. I personally found it was a wonderful session actually. I must say I personally felt proud of being able to be part of this eminent panel of people. Actually, they are all part of our family, and I really learned quite a lot, and I salute all of them. And thanks everyone. Of course, special thanks to Sam and also Atul. Atul, I really salute your boldness and brave speech that you gave, and you're so confident. And thanks everyone for joining, and also thanks to Arunji, Suguetan, and Sridham for hosting it. I found this to be one of the brilliant session actually.
I forgot, Sri Ramendran is still there.
Ramendran Chetan? Yeah, good.
Thank you. Okay.
Everyone.
Okay.
ചേട്ടാ വളരെ നന്ദി വന്നേന്. Thank you.
Okay, Arun. Okay. അപ്പോൾ നമുക്ക് അടുത്തതിലേക്ക് കടക്കാം. അടുത്തത് മേ ബി ഓവർ ടു ശ്രീറാം.
ഞാൻ നമ്മുടെ റെസിഡൻറ്റ് എന്ത് മ്യൂസിക്കിൽ വന്നു കഴിഞ്ഞാലും നമ്മുടെ റെസിഡൻറ്റ് എക്സ്പേർട്ടാണ് ബാബ് രാജേഷ്. അപ്പം മാഷിനെ ഞാനിതാ ഇൻവൈറ്റ് ചെയ്യുന്നു. അൺമ്യൂട്ട് ചെയ്തോളൂ.
താങ്ക്സ് ശ്രീറാം. കേൾക്കുന്നുണ്ടല്ലോ?
യെസ്, ഞാനൊന്ന് യൂട്യൂബും കൂടെ ഒന്ന് ക്ലോസ് ചെയ്തോട്ടെ.